audience. I'm happy to welcome you to our today's webinar. The topic will be specific biomarkers for lysosomal storage disorders, diagnosis, prediction, and therapy monitoring. I welcome our Dr. Claudia Cosma, the Vice President, High Throughput Testing and Biomarker Development at Centogene AG. Claudia Cosma will first introduce you to the company, then talk about metabolic diseases like Fabry disease and Gaucher disease, as well as other lysosomal storage disorders. Last but not least, she will present the development of biomarkers and especially novel biomarkers at Centogene. Dear Claudia, I would like to hand over to you. Many thanks.
Hello, I would like to speak you a little bit about Centogene and why we can have a lot of knowledge to show to other people. We are based in Germany. We have 5 affiliations worldwide, over 280 employees from 42 different countries, and we receive samples from over 115 different countries. This year, in the first semester, we received over 60 samples and process them in Rostock. We have a large amount of different techniques that we are using in our laboratories. We have Sanger sequencing, NGS panel, full exomes and full genome sequencing. Everything is coupled with biochemistry where it's possible. We have copy number variation and mutation quantifications, Cento and IPT. Everything is enabled via our Cento card, our simply logistical solution, which is a filter card on which we can receive samples from donors at room temperature from all over the world. All data that we can share, we dump it in CentoMD to be available for other people to use it. I would like to speak mainly today about rare diseases and especially lysosomal storage diseases. We are a private laboratory and we are working with patient samples and under pressure of time. We try to do as fast as possible the diagnosis, but in the same time we try to do it at the highest quality possible. Our clients and especially their physician can contact our medical and quality team. Sample can be received using our Cento card. The Cento card, the sample and the processing that is taking place can be followed up using our clinical data management, especially Cento portal. The diagnostic is performed as fast as possible with very low turnaround times. And the reporting is released back to the client via our clinical data management. And if we have a consent, we can put the data, anonymized of course, in CentoMD for other people to use it. Why rare diseases? If you take in consideration only one rare disease, there are just a few patients, but if we collect all the rare diseases out there, actually over 350 million people worldwide are suffering of one disease or another, which makes actually the third most populous country in the world. Half of these people are actually children. About 30% of these children will not live to see their 5th birthday, and for most of the people, average diagnosis time, especially in US, is somewhere between 7 to 8 years. 80% of the rare diseases are actually caused by mutation in our genomes. From all these families of rare diseases, I would like to speak about lysosomal storage diseases. In a normal population, a normal healthy persons, all the molecules are processed and recycled in the body using lysosomes. These lysosomes are organelles in our cells. encapsulated in a membrane and have a mixture of different enzymes, exoenzymes that are cutting sequential big molecules in much smaller molecules to be recycled in the body. If one of these enzymes is not working, we're going to have a proliferation of these lysosomes, an accumulation of the substrate for that enzyme, and the cells are expanding until severe clinical symptoms are visible. If this disease has an early onset on children, this can be seen also in bones, but also in soft tissues. If that is happening later in life, we're going to see mainly in the soft tissue, and especially in internal organs such as liver and spleen. So how do we receive samples at Centogene? We receive samples from doctors that recognize one of the symptoms, such as hepatosplenomegaly, physical or mental retardation. We receive samples from incidental findings in clinical tests. For example, newborn screening with high glycosaminoglycans in urine. We receive samples with a family history of LSDs or with unexplained infantile death. These are cases with a suspicion of LSD. When the samples are arriving here, we are performing biochemical tests such as enzymatic assays or biomarkers. We confirm the results directly with a genetic test. And what is very interesting is that it's not necessary to receive a second sample. Everything that is done in one step within 10 days. In the end, we are also, for specific diseases, we are able to follow the treatment and the evolution of the disease using biomarker level in the body. Usually a diagnostic diagnostic of LSD is performed in the following steps. We receive a sample here, either ADTA blood or dry blood spots, usually dry blood spots. We perform the enzymatic test and a control enzyme to show the validity of the experiments. After that, if the enzymatic test is normal, we send a report. If the enzymatic assay is pathological and or the control enzyme is pathological, we go to respective gene or genes sequencing. If if enzymatic assay and the genetics are in concordance, we send a pathological report. If the gene is gene sequencing is clean and the enzymatic test show the pathological results and the sample is valid, we go further to MLPA or qPCR to find about the deletion duplication in the genes. And then we send the report. Why biomarker? On one hand, we have the clinical information, and the clinical information a lot of time is subjective. What for a doctor means a headache, for another doctor could mean a severe migraine. On the other hand, we have the genetic variants. These are very rare diseases. A lot of the mutations that we found or variants that we found are with unclear significance. That means we need a way to quantify the clinical information and genetic variants and to find a way to explain them. And that is the role of biomarker. A biomarker has to be able to identify the risk for to develop a disease, to assess the disease severity if the disease is already there, to predict how this will evolve in the future, and if it's possible to guide the treatment. If two of these points are met, then we have a good biomarker. If all the four points are met, we have a perfect biomarker. When we speak about biomarkers with different persons from different fields, we're going to have a different understanding. If I'm speaking with a doctor, with a physician, an enzymatic activity is a biomarker. If I'm speaking with somebody from chemistry, a protein, especially the release of some protein in different events will be also a biomarker. If I'm speaking with a genetician, they will tell me that a mutation is a biomarker, and everybody is right. For me, what a biomarker means is actually the metabolite, the phenotyping at the molecular level, if I'm allowed to say this. For example, in the case of Fabry disease, Gaucher disease, Niemann-Pick, sphingolipids. Usually, a quantification of biomarkers will be done in the lab using targeted MS. What that means, mass spectrometry, and especially multiple reaction monitoring, mass spectrometry looking for something that we know is already there. We add to all the samples an internal standard, which could be any compound, which cannot be found in the original sample. And then we use it as a landmark. And compared to this internal standard, we can quantify how much an analyte is in the sample. Usually, the two approaches, enzymatic assays and metabolite approach, will have advantages and disadvantages. On one hand, for the enzymatic assay, we make an extraction from dry blood spots. We incubate with a substrate. We can put as much substrate as it allows us. That means we're going to obtain enzymatic product as much as we can detect with a different analytical methods. That could be fluorimetry, spectrometry, mass spectrometry. Again, the advantage besides the quantity that we can detect will be the quantity of the analyte that we can detect, could be also the tradition that LSD were actually, enzymatic assays for LSDs were developed even in the 70s. On other systems, on a huge amount of samples, of course, but still they are around for a long time. Substrates are also around for about half of these lysosomal storage diseases. Disadvantage for enzymatic activity would be that enzyme stability is a problem, especially if you receive a sample from Saudi Arabia or from Kazakhstan, from Rio de Janeiro. So during the transport and the storage time up to the analysis, the stability could be affected. Another disadvantage is that once the enzyme replacement therapy starts, we cannot use this assay as a biomarker. Now, if we go to biomarkers, we simply extract everything from blood, and we inject everything in the mass spectrometry. The analytes reflect exactly what is happening in the body. Whatever is happening in the body correlates exactly with the quantity of biomarker that we measure. Requires very little amount of sample, and determination takes place directly without needing expensive substrates or customized substrates for this. Disadvantages are that these are relatively new approaches. That means you're gonna need, we are just at the beginning of this field. Another disadvantage is that being such a little amount of analyte in the sample, you need very well-defined analytical methods and very sensitive, such as multiple reaction monitoring. But the best way to do a diagnostic is actually to combine the two of them. If from the biochemical point of view, you have both enzymatic assay and biomarker determined, then it's quasi a finalized diagnostic. You just need a small confirmation or mutation confirmation. Now, I would like to speak about two different diseases, the most prevalent ones. One is Gaucher disease and the other one is Fabry. On one hand, Gaucher disease is an autosomal recessive disease. That means carrier should not present any kind of symptoms. Pathological cases should present at least two mutations on two different alleles. This is an important fact because this is the main difference between the two diseases, and especially in the diagnostic area. Typical symptoms, growth retardation, moderate to severe organomegaly, bone diseases, bone pain, anemia, and other blood-related symptoms. At the molecular level, we can see a hallmark symptoms, which is the Gaucher cell, very enlarged cells because of the accumulation of these metabolites, which are not being processed by beta-glucocerebrosidase. Now, Gaucher Centogene. We could receive three types of samples for diagnosis, either plasma, and we perform lyso-Gb1, which is our biomarker for Gaucher disease. And if it's normal, it's normal. If it's pathological, it's pathological. We could receive EDTA blood with specifically request for lyso-Gb1, or we can do a classical stepwise diagnostic, which can be, in the first phase we have glucocerebrosidase enzymatic test, followed by, together with a control enzyme, followed by biomarker quantification in dry blood spots and GBA gene sequencing. In the end, everything has to be done within 10 days, or at least this is our target turnaround time. In case biomarker and enzymatic assay are pathological, but GBA gene sequencing is not showing any kind of mutation, we have to continue also with qPCR. Especially because this gene is a long-range gene and it's a little bit difficult to detect some of the mutations. An overview of identified cases at of Gaucher disease at Centogene. We receive samples, as I already said, from a lot of countries in the world, but mainly from Middle East, Europe, and Latin America. And because of that, the geographical distribution here is not as much epidemiological distribution, but more reflects more or less the number of samples that we receive from different regions. We have, up to June this year, 649 cases, real pathological affected cases, and inclusive with carriers, we have identified over 1,000 Gaucher individuals. If we think about the age of diagnosis, so the age when we receive sample, we can see that for the pathological cases, actually they are not in childhood, but mainly we have a very large range from childhood to adulthood with an average of 24 years. For carrier, even longer. Depending on the region, also the age is different. We have a different mutation. We see different mutations in different regions. Most of them in GBA are substitution and simple missense, but doesn't mean splicing, frameshift, or nonsense are not present. From all the mutations we found, we saw over 1,300 variants, and from those, most of them are published. About 74 were not published. These are all the variants we found. We found over 150 unique variants, from which only half of them are published actually. The other half can be found actually in CentoMD. If we look up at the enzymatic assay, we tried to put the cutoff S to the highest sensitivity possible even if we sometimes we lose specificity. We go with a very high cutoff to be sure that during the we can cover the transport time and the preparation time, and to identify these Gaucher patients. If we compare with other lysosomal storage diseases, beta-glucocerebrosidase has a different range for Gaucher patients, other LSDs, and normal controls. With our assay, we can actually catch some of the carriers. For us, as I told before, we could have either perfect biomarkers or good biomarkers. In our case, our star biomarker is glucosyl-sphingosine. It hits all the four properties that we are looking for in Gaucher in a biomarker. We have sensitivity and specificity of 100%, and we can clearly distinguish between patients and controls. As you can see here, the scale goes up to 3,000, which means compared with the cutoffs, more than 300 times higher. Considering only the homozygous mutation, the patients with homozygous mutation, which in this case is over 185, you could see here that for different mutations we have different levels of lyso-Gb1. Some of them, for example, if you look up here, 1226, these are cases of adults with very mild symptoms of Gaucher, while sometimes in this case this is quite a high biomarker, and also it's correlating with the phenotype. The child could not survive the first month after birth. This biomarker can be used also for follow-up studies. This is a set case, a very sad case from a few years back. A known family of Gaucher had another child. We received samples collected in utero from this fetus, and we measured, and it was already in pathological range. After birth, we measured every 2 weeks another measurement of lyso-Gb1, and the lyso-Gb1 increased severely. At one point, where the arrow is, transplant of bone marrow was performed. We had a small drop in lyso-Gb1, but the disease evolved quite fast back on track, and the child expired after 6 months. We have also some better cases. For example, this is a patient diagnosed at 10 years old with a quite common mutation. We started with a 160 nanogram per milliliter lyso-Gb1 with a very low dosage of enzyme replacement therapy. The lyso-Gb1 dropped quite fast to quasi-normal levels, and then starts to accumulate again. The clinical symptoms reappeared only when after a few visits where the lyso-Gb1 could be seen increased. The doctor increased the enzyme replacement therapy dosage, and then now we could see a reduction of lyso-Gb1. And now the patient lives quite normal life. Another case, a 17 years patient, they started already with a high dosage of enzyme replacement therapy, and they went stepwise with increment until they reached a level where the lyso-Gb1 could be considered normal, and the symptoms disappeared. And now we still monitor this patient. Those were only single cases. Now I can show you some of the clinical studies we did. For example, some treatments with velaglucerase. The treatment was applied to a group of 22 type 1 Gaucher, and we see until the end of the study, so after more than 200 weeks, we see a reduction of, in average, of 82% of lyso-Gb1, and we see also the phenotype reflecting this. On the other hand, we have also another clinical study for a switch group. Patients who had received for at least 2 years previously to this switch to another enzyme, a treatment with imiglucerase. And we see a reduction up to 52% compared with the baseline. We try to see if this lyso-Gb1 is in the pathological or pathophysiology of Gaucher. We took mice, healthy mice. We injected subcutaneously with lyso-Gb1, and we saw a decrement of hemoglobin and hematocrit, and a severely increased increment of spleen. Practically, we provoke symptoms in Gaucher mice with symptoms of Gaucher in otherwise healthy mice. This leads us to believe that for us, lyso-Gb1 is an ideal biomarker. It's linked to a clinical manifestation. It's easy to quantify in peripheral blood or in plasma. Easily accessible with a clinical sample, realistically reflects what is happening in the body, and reflects the phenotype very well. Reflects also the therapeutic measurements, and elucidates molecular pathogenesis of the disease. In Fabry disease, on the other hand, this is a X-linked lysosomal storage disease. It's a deficiency of alpha-galactosidase A, and is characterized by accumulation of GB3 and lyso-Gb3. The frequency is reported to be somewhere between 1 in 4,000 and 1 in 12,000. About 1% out of all young stroke patients between 18 and 15 years are suffering of Fabry. But what is very important is that full-blown picture could be a symptom of stroke, cardiomyopathy, nephropathy, corneal symptoms, angiokeratoma, hypohidrosis. But the majority of Fabry phenotypes are monosymptomatic or oligosymptomatic. What that means, that full-blown picture of Fabry will be only in about 10% of the reported cases. All the others are either oligosymptomatic or monosymptomatic. And lyso-Gb3 reflects exactly what is happening in the phenotype. If the patient is monosymptomatic or asymptomatic, we have a very low lyso-Gb3. If the patient is a full-blown picture, has a full-blown picture of Fabry, will be also reflected in the level of lyso-Gb3. In a clinical study we did, we showed that contrary to previous known data that most of the Fabry patients can be found in population that have dialysis, we showed that most of the patients that we find are actually in the stroke population under 15 years old. GLA mutation, from all the mutations that we discovered here, we sequenced over 76,000 alleles. We found about 3,060 300, 600, oh, excuse me, 3,600 pathogenic alleles and unique variants over 500. If you can see, most of them are missense. How do we do diagnostic as Centogene? For Fabry, because this is X-linked, we have to segregate males from females. For females, we start screening with GLA gene sequencing, and we confirm it with Lyso-Gb3. For males, we go with classical stepwise diagnostic, GLA enzymatic activity. We go then to Lyso-Gb3 and GLA gene sequencing. GLA enzymatic assay also presents a few problems. Some of the males, mild males or late offset, will present a normal enzymatic activity, and as well some of the females. To compensate for this, we are performing in parallel with the GLA enzymatic assay also a beta-glucuronidase. This enzyme is co-localized with alpha-galactosidase, and we always monitor also the ratio between GLA and beta-glucuronidase. Lyso-Gb3, as I already mentioned, will reflect also what is happening in the phenotype of the Fabry. Some affected males are just monosymptomatic and will have a very low Lyso-Gb3. Some of them will present very high Lyso-Gb3. For those that are very high, you see here listed a few most severe mutations that we found, and considering that our cutoff is at 1.8, you can see that we Lyso-Gb3 levels could reach over 200 nanogram per milliliter. We use Lyso-Gb3 in Fabry for monitoring, and if you remember before, Lyso-Gb1 has a spectacular decrement when enzyme replacement therapy is administered. In the case of Lyso-Gb3, we have a very slow decrement. In a few years, we can get from 10 to 2, or in this case, from 70 to 20. In other case, without treatment, we can also see an evolution of the disease, and this evolution is taking place also very slowly compared with Lyso-Gb1. That is not the only those are not the only biomarkers we have. For example, for Niemann-Pick, A, B, and C, we have lysosphingomyelin 509. And you can see here for NPC cases, for example, a complete separation between normal controls and pathological. Of course, we have to think about discovering new biomarkers. And while in diagnostic, we are always using triple quad mass spectrometry and multiple reaction monitoring mass spectrometry. For discovering of new biomarkers, we have to go untargeted. What that means, we have to do full metabolic profiling for the samples we are investigated. We have in our biobase samples from a lot of patients. We have large cohorts. And from this, we have the blood, we make an extraction, and we inject everything in ion mobility cutoff. With this, we have 5-dimensional data that we can actually compare to identify whatever sticks out in metabolomics of the lysosomal storage diseases compared with healthy individuals. Although the scheme looks very simplistic, the workflow is a little bit more complicated than that. Our multidimensional data will be loaded in a statistical analysis program. The total ion chromatogram are aligned, and using a large number of vectors, all the samples will be overlapped and mathematically correlated. Using an intensity ion map, we're going to see how good the overlapping and the vector editing is, and we can actually obtain an ion map. From there, we normalize to the reference to be sure that we're going to have perfect quantification. And from there, we manually analyze all the candidates to see the differences between the normal cohort and the pathological cohort. That led us to development, for example, of another biomarker for Fabry disease. We just published it this year, and it's about Fabry disease. Fabry disease is characterized by an acidic ceramidase deficiency. By measuring a lot of ceramides, we realized that only long-range, very long-chain ceramides are accumulating, and especially ceramide C26. At a better investigation of this finding, we saw that we can see two different isomers or isoforms of this ceramide, one of which is a trans ceramide, while the other one is, in our opinion, that is a cis ceramide. If this is true, that is the first time when a cis ceramide is reported in a living organism. The two isomers are perfectly characterized by an exact mass, a fragmentation pattern, and a collision cross-section. What is interesting of this is that although isoform 2 have no cannot be used to distinguish between normal and pathological samples, when we look up actually on the first isoform, we can clearly distinguish both in plasma and in dry blood spots between the pathological and normal. And when we Fabry disease is an ultra-rare disease. Up to now, only about 100 cases were reported. In our biobase, we had the 10 cases and 10 carriers, and when we can see this, we can actually separate them on newborn, juvenile, and adults, and we can see the distribution of two isoforms and the prevalence of these isoforms. So this concludes my talk. I'm open for discussion. In a world where treatment options are developing continuously for rare diseases, if you don't look, if you don't think, you don't look. If you don't look, you do not find. If you do not find, you cannot treat. And in my opinion, biomarkers are the easiest way to do that. Thank you very much.
Thank you, Claudia Cosmo, for this very interesting presentation. I have one question from the audience, and this is, are biomarkers enough for diagnostics or only for screening? Maybe you could say something about it. Thank you.
Depends on the biomarker. If we consider lyso-Gb1, one can use this for diagnostic, not only for screening. In the case of lyso-Gb3, the biomarker is a method of screening and an aid in the diagnosis process. In the case of lyso-509, again, this reflects exactly what is happening in phenotype and can be used for diagnosis.
Many thanks for this reply. As far as I see, there are no further questions for now. The audience can feel free to send some questions to the host, Centogene, or Annemarie Neumann. But if there are no further questions coming up, then I would like to thank you, Dr. Claudia Kosma, and would like to tell you as well that you can send all the questions that might come up within the next hours to our customer support or to webinars@centogene.com. Many thanks.
Thank you very much.